Is this supposed to? So this is not working anymore. Oh, it's working, okay. This is the lab. Actually, Nima and Maddie are here. If you could stand up, or there you go. So imagine if you could obtain generalizable insight from just a case report or a couple of patients, or unveil outlier biomarkers for super responders or those that have severe events, adverse events to therapy, or unresponsive patients. Imagine if you could swiftly launch a small and statistically powered clinical trial. How many of here are clinicians? Oh, okay, a handful. Among clinicians, I'd like you to, there's a heartfelt feeling for the clinical trialists. It's one of the hardest scientific career because, especially starting nowadays, you start and you're probably requiring to come up with innovative ideas where all low-hanging fruits have been taken on frequent diseases. Otherwise, you won't be able to obtain a cohort large enough in 5 years and sometimes 6 or 7. And you've got to pick a couple of choices, and you have those 3 seminal papers that either make it or don't for your tenure. So the first 5 or 6 years are very arduous because you're not yet renowned. It's hard to obtain the grants for these clinical trials, and you're stuck in grooves that have been where a lot of people have walked before, because if you were to go for infrequent diseases or stratified complex diseases that are frequent, you'd come down into statistically underpowered conditions. So this is something that was close to my heart, and I was wondering, how can we help this kind of studies? There's also, how many of you are familiar with when a physician says idiopathic? Yeah, so the idiot is the physician, right? That's what I was told when I was trained. That means we don't, it looks really serious in front of a patient or family members, but actually it means we don't know the cause. And I still remember 20 years ago when the genome was, or 25 years ago when genomic studies were coming out, and researchers would come and would say, well, it was $4,000 per sample to run, and we're speaking of 20 years ago, to run these 2 samples. Could you tell me what's the results here? And I was thinking, you know, where are your measures of dispersion? And then of 1, 2 condition, very difficult. A full change would give you a lot of errors, and I thought, you know, they're out of their mind. You need multiple measurement in each condition, but actually the idiot was me, because thinking about it deeply with some of my colleagues in the mid-2000s, we realized we actually had statistical methods that could approach that, and single subject studies in the omics area became possible from 2 samples rather than from series. So statistics over repeated measures over time can give you in the patient some p-values, but that was one of the question. So the third point here was about the farmed and discovered areas for drugs, or the problem I was speaking about clinical trialists. And I don't know how many of you are aware that there's just about 3,500 compounds that the Food and Drug Administration has accepted to treat. Out of that, the combinations and the presentation and the form and the way we sell them, there's 100,000 unique identifier in the US to sell those 3,500 compounds, but they only address about as many conditions. And we know 65,000 diseases, of which only 8,000 are Mendelian, single gene disease. So by far, and you know, arguably single gene diseases, the solution could arise elsewhere in targeted therapy, gene editing, and things like that. But for the others, these are complex conditions. So we're probably just treating 4% of patients. And we clinicians, when we're trained, we're never told that. We're trained with our, just like a horse with their, how do you call that, blinders, right? They're blinders. And what we realize in practice that there's the frequent disease occupy our clinics, but when we scratch a surface of that, for example, in pediatrics, in a referral to subspecialty in pediatrics, 40% of patients have no drug to treat them. We can only treat symptoms. So coming back to how could we be disruptive? there's probably a micro paradigm shift we could do in clinical trials that are amenable to intermediate measurements at the biomolecules level if there's biomolecule dynamics, whether it's mass spectroscopy, proteomics, metabolomics, transcriptomics. Since by convenience we had access to transcriptomic, we'll show the aspects in transcriptomic here. And this is the number of adverse events that are described in clinical trials and reported. And we're in the area of 400,000 a year right now. So when we think of outliers in clinical trials, there's a lot, and there's a lot of opportunities if we're able to micro-stratify even in large clinical trials and understand what's going on in these individuals. So I'm trying to make a strong case here for considering micro cohorts. This will be the plan. I'll look into additional problems, the motivation, the system by which we use single subject studies, pull them together and beat the statistics of conventional cohort-based trials. We call that inter-N of one because it's between several groups of N of one. And then we'll look into machine classifiers and its imitation in future studies. That's the rate of failure also of clinical trials. So not understanding what's really going on, we're sometimes missing subset of cohorts. There's numerous examples, one in cardiology from Harvard, I can't remember which drug it was, that was rescued when they started looking into a subset of the cohort that were responders, while the majority were not. And originally they had balanced it as a failure, but they realized there was a subset that was responsive. Those were the statistics, though, that there's just 4% of diseases that are currently treated. This is simply, and I'm speaking to the choir here, systems biology or systems ways to look at problem. For example, prolonged bleeding time. We can go back to what we've studied in terms of intrinsic, extrinsic pathways. I've highlighted here different type of hemophilia in each of the proteins. Loss of function would give you a different number, 12, 12A, et cetera, right? But there's acquired form of prolonged bleeding time. And a good example is this gentleman at the bottom here with cirrhosis and alcoholism, with a reduction of protein expression without any mutation or loss of function of 4 proteins highlighted in red. And now you have obviously not the same plethora of phenotypes as you would have in hemophilia, but you have a dysfunction at the systems level that looks pretty much as like a prolonged bleeding time you'd have in a hemophilic person. So in other words, there's missed opportunity if we don't look at the system. And I didn't show you the data, but the NIH has funded over $2 billion of biomarker studies over the last 20 years. There's only about 20 biomarkers that were FDA approved during that period of time. We're speaking of, for the majority, single gene biomarkers. Now let's take a step back. We know 25,000 genes and 65,000 diseases. There's a problem there, right? So I think Einstein said something to the effect of applying the same, trying the same problem over and over, or the same solution over and over again, observing a problem. It's systems-based approaches are not well received at the NIH because we have a very large cohort that review us that still have a model that is mathematically easy to understand if you're not trained in numerically, like in applied physics, maths, or engineering, which are linear models. But clearly, just the suggestion that biomarkers would explain treatment of complex inheritance, which we cannot understand through a single gene, is a bit ludicrous when you think about it. So there are many ways in which we can already do single subject studies. We could compare, I think I have a pointer here. I'm not sure, on the top, oh, sorry. On the top, there's a reference genome against which we can assemble current genomes and have variants or mutation predictions. At the RNA-seq, you could do a reference transcriptome, but there's several problems with that when you think about it. The same applies for the proteome and the metabolome. First of all, people have different genetic backgrounds, so they could have totally adapted transcriptomes that are distinct for an environmental condition. So from the get-go, the baseline could be different and totally non-pathologic, right? Even twins, if I had a twin that still lived in Montreal, exposed to the winter over there, I guess when I was in Arizona, it was, but I lived in Arizona for a while, right? We may have a basal transcriptome that differs. Then each cell type probably has different basal transcriptome. So it's hard to make references. And we thought that being ourselves a reference to would be a better control condition. And when you think about it carefully, that's how we design cell lines. That's how we design many animal models, mice models. They're isogenic, with the exception of the gender, they're really isogenic. And it's very powered. For comparing two transcriptome in mice with conventional statistics, it takes 3 miles per condition, and you can get p-values at false discovery rate for a few hundred genes. If you do humans that are obviously heterogenic, it will take about 30 people per group to do these kind of comparisons. If we flip a human being into a single subject study, each human being is in a study that's isogenic. We're very powered. So what if we're able to predict without triplicates in cellular models or animal models the transcriptome differences? And that's what we've done. So only one observation in each condition. It's really a data scientist nightmare. The tools that are commonly available require replicate sample to calculate a measure of dispersion. Often you've studied that as the variance, right, or the standard deviation. But there are other methods to look into measures of dispersion, and I'll come to that. And the restrictive assumption of these statistics is that if you have two conditions without replicate, it's not eligible for those statistics. You don't meet the criteria of comparison. NOI-seq, DE-seq, DEG-seq, these are systems that will give you differences for a transcriptome over groups, and they all claim they could work in one condition, have never shown the results. We have a paper by Samir Rashid Zaim at the bottom here that compares all of these and show how 5 of our methods do better than they do, and it doesn't scale down when you're past three samples in a repeated measure in each condition. So I think I already discussed the motivation here. So if we look at the literature, if you want to look at an external database, we and others have developed methods on the top that look at an affected sample against a cohort reference. I've already discussed the limitation of that, but oh, okay. Instead of the pointer, it changes. Okay, you get it? The top one. Okay, thank you. Where were we? So, and you can find differentially expressed genes or differentially expressed pathways by these methods. In comparing affected sample versus a control in one subject, infusing external knowledge base, you have different techniques to derive differentially expressed genes. These are not very good. That's the paper I've pointed to of Rashid Zaim. And then differentially expressed pathways, those are have higher accuracy, with the problem now that your biomarker is a multi-gene biomarker, right? A multi-molecule of life biomarker. And what we're trying to transform is these large cohorts of case control, or this one here in which paired samples in one patient, let's say before and during treatment, are still require large cohort by a single subject and obtaining a statistic here. So that's the problem we're looking at. Rapidly, all of you are familiar with fold changes, right? If we have values for one molecule of life in two conditions, we divide the value in one condition by the value of the other condition. And if the did not affect the outcome, normally that should be around 1 plus an error rate of measurement, correct? The log of that, log of 1 is 0, gives you a normal statistic that's centered to 0. It's pretty nice statistics. So fold changes, if you look at it and you do an absolute value of your fold change, you now flip, let's say that something has reduced the expression by half, the other one has increased by half, and you have two molecules of life. One would have a fold change of 2, the other one of 1/2. and you do the log 2 of that, you'd get the results of -1 and 1. If you do the absolute value of that, then you're flipping all of your downregulated genes to with the upregulated genes to the same result after log transformation. So now you get more results in the dysregulated gene area or dysregulated transcript, and the unaltered ones nearing 0. But since you've taken no more curve and you flipped it, it's called a flip normal. And that's the type of statistics we're using to have measure of dispersion using mixture model. How many of you are familiar with statistical mixture model? It's really a relatively minimalist approach if you look at just 2 distribution in which there's very few parameters involved. And that's why we preferred that over 3, which would have been without the fold change. We could have looked into downregulated genes, upregulated genes, and unaltered genes. But having less parameters better and more powered. And we've done the analyses for that. There are other approaches to the fold change. You could look into a non-parametric statistics. The Wilcoxon works as well, but it's not as powered. You could look at Mahalanobis distance, and that also works. And we've used also clustering. You could try to cluster these fold changes in 2 groups. I'll just get to the results. So if you represent in these pathways that are altered, well, I forgot to tell you something, I'm sorry. Afterwards, you could look into this list of altered genes, the dysregulated ones here, infuse a knowledge base of which pathways they're part of and see whether there's an enrichment, often called also overrepresentation. The Fisher exact test is one example of statistic that could be used there, or hypergeometrics, a bit more precise. And then you get a p-value that you can correct by false discovery rate for each molecule of life, for each pathways. And here what you have with mixture models is a posterior probability, which is not the same as a p-value. So this is a bit superior in terms of what you can call correct or not. You get differentially expressed pathways and differentially expressed and you have the p-values associated with that. So you have the effect size of the pathway in terms of overrepresentation. That would be an odds ratio. So if each pathway is represented in one of the radial area of this representation here, we're informatician. It's not just making the computation. We want human beings to understand that. And I'll show you how a clinician could understand that in 5 minutes. I hope you see a pattern difference between the top and the bottom. If something is unchanged, the green expression goes to the medial area here. You don't see the gray, just like these guys here, maybe not this one here. And that means pretty much at the pathway level, unaltered odds ratio, the pathway is not altered here. And if you have overrepresentation by 10,000-fold, it will be the full radial area, like these areas here. And abrogation will be no color. Each of these wheels is one distinct patient. We've looked into the most altered pathways between the disease-free survival in more than 5 years and those that had death of disease and TCGA lung cancer. There were about 50 patients there. And those were the most diametrically extreme group. I think a clinician would find the pattern recognizable immediately. And what does it correspond to? Well, those chromatin assembly defects are right here. And we can see that's overrepresented in those that die of disease in less than a year, as well as the chromosome localization. Abrogated are hormone secretion and homeostasis. And those that survive more than 5 years, you can see they were nearly unaltered in these conditions. And what were we comparing? We're comparing lung tissue versus the margin of the lung, that's less affected by the cancer. Right? It's considered, it's not normal tissue, but it's not cancerous tissue. It's not normal because the cancer transforms its environment, which is the host. Yeah. These are from gene ontology, so we're not naming these. We're not naming these collections. These are the super classes in which these Go terms belong. So these are gene ontology molecular processes, and they belong to a super class that's here. I could even have put a Go term here or here. So all of this is data-driven. We don't do, there's curation. People that have created gene ontology. Now, most of you must be familiar with reactome gene ontology, right? This is, okay, so there's a large group of curators that have established that. It is biased in a certain way. It had to be studied. There are studies that show that those biased curated knowledge bases like PFAM, KEGG, and so on, probably map in cancer less than 50% of what appears to be clustering in terms of behavior and molecular behavior. So we don't know, there's a lot we don't know, but for what we know, there's that. There's unbiased way to approach it as well with yeast-2 hybrid, EQTL. You can create clusters of molecules that have similar behaviors from large databases and come unbiasedly and use these collections of groups of biomolecules to make your enrichment. So what I've shown to you is one that's based on gene ontology. We regularly use OMIM or KEGG, and you could use any of these. We also use EQTL regularly, microRNA targets regularly as well, yeast-2 hybrid. This is just to show you the how we calculate the odds ratio so that you're familiar with the effect size, because, you know, if we were to take 2 classrooms of students at the same grade in 2 different cities, there could be a half-pound difference in average weight between them. And it could be statistically significant, but clinically, I don't think there's anything we could address with a half a pound of difference. So the effect size is important. It's not just a p-value, but the effect size is important. In this case, we're speaking of overrepresentation, and we're looking at the gene set of the pathway, the genes assigned. So someone else is pointing, but me, right? It's funny. There's a red dot that started moving here. Interesting. I don't think so. No, it's here on the screen as well. The mouse is probably, I don't know if the mouse battery. I had a colleague that had a joke like that once. He thought his pointer was working, but there's another colleague that was pointing for him. And eventually he started moving all around. I don't know. So I see it on the screen as well. So I know it's not a joke. It's funny. So clearly when I press this button here, it activates something else. Maybe there's, so there's a gene set in each pathway. It's a bit, I think I'll stop using this. And if the red, so clearly something's going on and it's not me. It's probably the battery of the mouse or. Anyway, so you can see the enrichment here as being, how many of you are familiar with odds ratio in contingency tables? Yes, 1, 2, 3. Okay, half of the class. So it's pretty straightforward. Let's just move on. By the way, the log of the odds ratio is also a normal curve, right? So it's a really nice statistic also when you start looking at comparing effect sizes. Thank you. So let's go into the intern of 1. How do we calculate that? I'm showing again, I like to show what existed. You could consider using general linear model. You could consider using t-tests and so on. And the advantage and disadvantages, right? Large cohorts here. This one, they need to be isogenic to have 5 subjects, but that's okay in our cases. But in our case, with intern of 1, we're showing it works even less than 5 cases. So there's probably this method, aligned rank transform test, pre-existing from 2005, that was doing relatively good result that could have been applied in micro-cohort comparison. So what does it look like if we have patients from 2 different cohorts, one colored mauve and one colored green? You apply, in this case here, we compare 2 different statistics. So you apply the fold change, a mixture model, in this case, NOI-seq, and we compared the results. But if you had a group A and a group B, so this is what's happening in each patient, right? In each patient, you calculate that, you get an enrichment, and for one patient, you get the contingency table, the odds ratio and the p-value comes out for this patient, comes out for this patient as well. This is an alternate statistic, and we've compared any statistic over time. Then you group your patients together for each statistic, and you can compare the 2 groups. So it's like doing Sincerely, it's really like doing a meta-analysis that you've seen in the literature in clinical trials. You do one meta-analysis for group A, one meta-analysis for group B, and then we've shown a new statistic by which you can compare the two groups. And that's here, that's how you calculate your variance. It's a bit tricky. So for each patient, you go pull the, in the contingency table, a measure of dispersion from the pathway, the pathway data. So you get a variance coming out of each single subject study, as well as a p-value, as well as an effect size, like 3 components. And you can use these, I won't go too much into detail, but to compare two groups. And then you have group comparison from meta-analyses of single subject studies, rather than a conventional statistic that would find the averages between patients by pulling them all together. And we're showing here that if there's a single, so comparing a group that has breast cancer, for which the oncogene that drives the cancer is TP53 versus another one for which is PIK3CA, and these were prior groups that were already published in top journals. We just studied them by resampling smaller and smaller. Where could we find the result they obtained in that larger study, and which they would not have obtained with their statistic, and where does it fall? So you can see that these are the sizes of the cohorts here, and we're starting to see a signal at the pathway level when we look into the statistical interaction. And this is more detailed data per size of cohort. And you can see that the general linear model has a large, I wouldn't trust it when you go from 0 to 80% of precision. Precision is the positive predicting value. That's what clinicians want to see. And here's the recolor sensitivity. But you can start seeing that here, 9 versus 9, it beats the models we're proposing. And there was one that was the interim of one using NOI-seq, a very well-published method that's non-parametric. Another one is mixture model. And you can see the mixture model is always a bit superior. But one could have used very more broadly known statistics as well. The idea is to not conduct the study on a single measurement or a single group of measurement in one condition, but to use 2 conditions. So you're pulling together a number of strategies to reduce your sample size. This is a prospective study that was done in valley fever. We were funded 5 years and 2 grants. And it's a clinical trial where we started. So we had this group here with 8 individuals that had the pulmonary form of valley fever, which is self-limited, and the other one had the disseminated form, which is lethal unless you're treated for a lifetime with an antifungal drug. About 1% out of 500 that's exposed to coccidioidomycosis in Southern California, Southern Arizona, Southern Utah, and New Mexico will have the lethal form. And some even with the antifungal are not treated properly. If you're African American, it's 1 in 200. It's still a mystery for me why it's increased in a group of individuals that have a larger variability over their genome than we do as Caucasian. Native American have also an increased risk, and that again, it's bizarre because they've been exposed to these in the region for a longer period of time. The cohort to discover the signal was 3 versus, these are very hard cases to find, correct? And you 3 patients there versus 8, and the validation was 3 versus 3. So we have one study that leads to the other study, right? You have discovered genes that get validated in the second study, and these are what we do. We first calculate the pathways that are enriched between 2 conditions. In this case, we had an ex vivo assay exposed to coccidioidomycosis lysate. So we take the bug that causes the disease, we create a lysate, we take the blood of these subjects, and we expose it ex vivo, and the difference of transcriptome dynamics when enriched at the pathway level with these statistics is different between the 2 groups. And all the statistics are shown here, like in an infographic. These are our results. This is an alternate study where we looked into single cell of prostate cancer that's circulating in patients. It's a different way to present the result here, and it was which pathways were resistant or sensitive between the group that was EZT resistant, that's one of the treatment, versus those that were EZT naive. And you can see the pathways that are involved are quite different in the resistant ones. And it's a slightly different representation in which what's above the line here is overrepresented, and what's below the significant but under, so dysregulated in a different direction. What about learning classifiers? So this is a prospective cohort in pediatric asthmatic patients, which were all severe. At the organ level, which is along, let me see how I'm doing in time. Okay, I'm good. So, and I don't know if I've dropped here. So these pediatric asthmatic patients were maximally treated. They were indifferentiable. So those that will have, so we're looking at those that will be hospitalized versus those that won't be hospitalized in the coming year. And they were indifferentiable after the year when you look at them in terms of weight, gender, lung volume. There's a pulmonary test, the pulmonary function, in which you can have a bronchospasm that's induced. So it's a provocation test. They were indifferentiable at the organ test level as well. And they were all maximized in drugs. So they were pretty much as controlled as you can have. And because the assay that we did was ex vivo, exposing them to the human rhinovirus, so the common cold. So in pediatric patients, 20 years ago, it was clear epidemiologically that 70% of hospitalizations were associated with the common cold for these patients. Later on, the last decade, it was shown to be causal, not just the theory that it's the last irritant that comes on top of the others. So there's a hyperreactivity of their bronchi to the human rhinovirus, which otherwise would give us a runny nose and a headache, 50% the chance, or otherwise no symptom at all. We only had 21 patients. So when I looked into the data, I told my colleague, Fernando Martinez, that had a dozen papers in Lancet and the New England Journal and changed the way we treat pediatric asthma. I said, you know, you've been sitting on this data for 4 years. Harvard failed to analyze it and so on and so forth. Give me a chance. And I came with the hypothesis I could take some Duke subjects, probably their students, that they had exposed to rhinovirus in vivo, and for which the data was available. So he told me I was out of my mind. I reminded him that those are also, his asthmatic patients were human, and that there would be some pathways that are involved in having symptoms. They're not asthmatic on the top, having simple symptoms of rhinovirus, which could help us learn a classifier that's fully specified. That's very important. Have the studies that have been published in the last 20 years, even in top journal like the New England Journal of Medicine, cheat. They cheat. I think they were just unaware, and even the reviewers were unaware. So they passed 250 genes, and then they relearn a new classifier in a new cohort. Please give me 250 genes found at random. I'll find your classifier on any of these data sets. So with enough patients, I'll find it. So that's the point, right? Fully specified, if you think of a linear model, it means that the weight by which you multiply the value of each of the transcript and that you add to the next weight times the next transcript is fixed. You don't change the weights. And then you have a threshold, right? Above the threshold, it's one group, and below the threshold, it's another group. It's a very easy way to conceive what you do. You don't cheat. You don't change these weights. You don't readapt the weight to the next data set. So that's what we did here. We did that, and it did work. So indeed, the asthmatic patients are human beings, after all. My colleague admitted that. Would we have had a cohort of asthmatic patients that were pediatric, we'd have had probably a better result. But this was one of a kind, because when you think about it, we're looking at a dynamic exposure and change between two conditions. So this is the first classifier in the literature that's based on genome by environment interaction. All other classifiers are actually on taking static result. They're not looking into the dynamic change, and it's just on the change that we're measuring the outcome. So this is, if you think of it, a provocation assay for the clinicians. We do that on patients all the time. Do we know if someone is neurologically deprived if you don't test the reflexes or the strength, right? So we provoke certain situations. We get more information out of a physical exam. So this is no different at the biological level. And this is what's showing you that they were no different. The group that had no exacerbation or recurrent exacerbation, you can look at the p-values here, are not significantly different in any of the conditions. These here are measured on the pulmonary function. And this here are each of the gene ontology terms. Now we've color-coded them to make it easier to see. These are the superclasses in gene ontology, molecular, biological processes. And you can see a color coding between our true positive and the true negative, and then between the false positive and the false negative. You can see we missed, right, in some area. One way that we can show you we're not cheating is that in all cases, do you see the purple is increased everywhere? It's totally useless. So that was useful. The classifier was fully developed on healthy subjects, probably students, exposed to rhinovirus. They were also exposed to influenza, by the way, and another virus. And we could show with the influenza, the classifier found didn't work. So if you don't have the right stimulus, it's not non-specific about any virus exposure, right? It's very specific to the rhinovirus. So you had like a positive control there. So this one here was designed in a classifier to discriminate among healthy subjects whether you would have a headache and a runny nose or not. And it corresponds to the innate immune response. It's totally useless in asthmatic patients, at least the pediatric ones, to find differences between group. But that shows you also that, you know, we didn't cheat. We used the classifier as it was, and it had components that were useless in asthma. And that was expected. On the other hand, you can see that acquired immunity and was the yellow here. Chromatin organization was overexpressed in each patient of the virogram, well, of those that were true positive, that were predicted would be hospitalized, versus abrogated in terms of pathways in those that were non-hospitalized. So there was probably a hyper response in the acquired immunity, which goes with the theory, right, that there's an hyper-responsive aspect of these patients that will be hospitalized. That's different. So biomolecularly, they're different. And by the way, genetic studies that look into asthma have not discovered predisposing genetic SNPs for asthma. There's very little result there. It seems the methylome at birth is already different between infant that will have asthma later on in life and those that won't. So those are part of the studies of Fernando Martinez. And we knew that genetics would probably not have been the right way to look for these between these patients, but functionally, we can see at the cell expression, this is happening. And now you can think of another problem. So what I hope you realize is that we are looking at 25,000 transcript and then 5,000 pathways, and look at comparison between that in the mirror 10 versus 10 patients, right? It's very difficult when you have a high number of variables and a small number of patients. Generally, the statistics will fail. It usually takes 100 to 300 patients in each group to demonstrate a classifier and validate it. And we're at 10 versus 10. It's a good 10-fold reduction of size. So what if we go even more, we're more outrageous in terms of comparison, and we're looking at pathway interaction. So we're looking at 5,000 pathways times 5,000 pathways, 25 million comparison. Could we find signal in pathway interaction? And that's really an enormous dimension for a mere number of patients. One of the hypotheses we had is that instead of looking into all these millions of gene interactions, or gene set interactions, we would look at those that are concordant between single subject studies, regardless of their group assignment. So that's not cheating, it's just before cheating. You're not looking at the group assignment. You say, wait a second, why should I look into a pathway or a biomolecule that's never changing between any patient, regardless of their group, regardless whether they'll be hospitalized or not? why do that, right? We could eliminate them from the get-go and just study what varies. So in traditional transcriptome analyses, what we do is a coefficient of variation. So the fold change divided by the expression between each transcript gives you a normalized fold change. The variance divided by the expression gives you a normalized variance between all transcript. And that's a way by which you can reduce the dimensionality of 25,000 transcript to something that at least varies in your cohort. And then afterwards, you look at the group assignment. So you're not cheating. So this is a similar way. We're saying we have pathway effect size and p-values in each patient. Without looking whether it's those that will be sick or not, we can say, an inclusion to the study of a pathway should be one that varies. Otherwise, why include it? So that's not cheating. And if you think of it, we have 20 patients, right? We tried to see, well, where would it be optimal? And we found that probably in this case, 12 out of 20. So we would only look at pathways. Well, we tried everyone, right? We would only look at, it's a proof of concept that had never been done. So we're trying to see where, do we have a sweet spot somewhere where it works? And when 12 patients out of the 20 have a variation, we don't care in which direction, we don't care in which group the patient is assigned, let's include the pathway. That goes from 6,000 gene sets to 98 gene set. And if you look at the number of classifier features and that's pathway interaction, 6,000 square is 36 million. For the engineer there, it's 36 million minus 6,000, right? Because we don't want to have self-to-self interaction, but above 36 million. And here we're just at 9,000 gene set interaction with this simplifying hypothesis. There's another one we've applied as well, which is a binomial random forest algorithm. I've developed an AI expert. So we're applying two strategies, and we don't have time to get in the detail here. Those that are interested, there's a paper on that we've published. And when you use the two of these, we actually get a signal. And what's more interested, remember I've shown you this graph before, correct? I've shown you chromatin interaction. Now, you're all biologists, you all understand that, well, what does it tell us about the transcriptome? So there's something, not expressed? Is it because the folding of the chromatin in this area is not exposed? There's something going on. So what we see is that chromatin interaction right here is interacting with pretty much every other goal term that was part of our classifier. So now we went from this representation in an area that should have been nearly impossible unless you have some simplifying hypotheses, and I've shown you one that made sense from the biological sense. And now we're able to even look at interactions between these pathways. So this is still in progress, but this is one of a kind. There's no pathway interaction computationally studied in the literature in an unbiased way, right? So in terms of the challenges, we're still working with transcriptome. We'd like to work into other conditions. We are working in metabolome in parallel. We've done a bit of proteome in the past. We believe that these single subject studies are applicable to questionnaire and even clinical data. It may not be the statistics we're using, but the idea of taking the same person into two different conditions and look at the difference of it, that's what clinicians do all the time when they practice, right? They're looking into panels that inform them what's going on, the dynamics, where they're going on. We're operating in the high throughput, taking knowledge bases from the outside and mimicking for patients what biologists or physicians are thinking, where they're using their knowledge and trying to interpret the result. The difference here is that it's done computationally at scale. So many more hypotheses can be tested than a human brain would be able to do. Obtaining tissue is rare. A substitute for all these methods where you could get single subject studies and actually interactions within a patient are the single cell analyses with large quantity of single cells in an experimental condition where cells are controlled with the other ones, for example, with CRISPR-Cas9 editing. And you could also look into CRISPR-Cas9 interactions if you did two genes, right? So that's an alternate way. I can see a number of ways by which One need not use the methods we've published, but it's definitely going to be an area that grows rapidly because of single cell analyses. Um, one of the limitations that's current resolution, the pathway level, and multi-gene biomarkers is not well accepted, but fortunately, from the Genome-Wide Association studies, there are new scores that are multigenic, that are becoming popular and are becoming mainstream, and I think it will help our community also at the dynamics of other parts of the genome to be accepted. And then when you reduce the subject size, you need to ensure that all their other clinical variables are comparable, because now you have confounding factors that come from individuality. Think about it. I've shown that we could do 3 disseminated cocci, dioidomycosis, valley fever, versus 8 to discover the pathways that were altered, and validate it in 3 versus 3, and it worked. But you need to have very well-matched individuals. Otherwise, what you get into are a lot of confounders. New technologies always bring new problems and new challenges, and that's where we are right now. Take-home messages, I think combining single subject studies require smaller clinical groups for comparison, so it enables smaller clinical trials. We call them micro cohorts. Again, think micro-stratifying large complex diseases for individuals that are rare. I'll give you an example. When I did that breast cancer study, you've seen that, there were 2 oncogenes that were driving. There were 50 patients there. There was not a single African-American. There were 2 Asians, I think. And if we look at the larger set in the what's called TCGA, the Cancer Genome Atlas from the NCI, they took the samples by convenience. They've asked, any samples you have, we'll do the transcriptome, the genome on it, and we'll provide you the result. And when you take samples by convenience across cancer scientists all over US that are well-funded, what do you get? White Caucasian women. There were 3 African-American among 500. 10% of the US population is African-American, right? There should have been 50 for proper representation. It gives information on who had access to cancer centers, who had the money to go there at the time, right? So on the other hand, these techniques could now triangulate and look into what's going on. I haven't shown you, but we can compare 1 to many. So if you have an outlier, we could compare it to the group and show what's going on there. Obviously, the group needs to be controlled for all the other components. Otherwise, you have these confounders that are also showing the difference. We don't call it, so whom are familiar with the curse of dimensionality? Well, okay, well, in data science, having too many variables and too few individuals upon which you calculate it, it's a curse of dimensionality. You're underpowered, right? You've heard that, underpowered. We call that a blessing of dimensionality, to have a lot of measures in one patient. In 2 conditions, that's the point. In 1 condition, it's a curse. In 2 conditions, it's a blessing because we get signal. So these are the funding areas and the support. Thank you very much.